Welcome back, everyone. We are now at Module 5 and we're going to discuss hormones and fertility. So this is actually a sort of a perfect flow from Module 4 because we're going to finish off the folate. I know we've talked about it at length, but there will be a bit of a section on folate in relation to neural tube defects in this, and then, believe me, I promise you'll be done with the folate. We're going to talk about factors that affect hormone and reproductive health. We are going to talk about key SNPs that can influence hormone balance and fertility. We'll touch on hormone receptors and talk about a gene that has been associated with thyroid function. We'll talk about the role of methylation in estrogen metabolism, and we'll also talk about oxidative stress and antioxidant-related genes. The learning objectives are to identify endogenous and environmental factors that can influence both our hormones and reproductive health, but just know that there will be more of a focus on this in the next module when we talk about detoxification, because that's when we'll really go into these environmental pollutants. We'll examine gene variants related to both male and female fertility issues, and to understand the functional role of key enzymes and their cofactors and how genetic variations can impact on hormone synthesis, hormone metabolism, and hormone excretion. We'll also review the role of methylation, again following on from Module 4. We don't really need to go into methylation in detail, but we'll touch on it because it is so important, particularly in relation to fertility, but also the metabolism of some hormones. We'll describe the impact of methylation and antioxidant-associated genes on endocrine health, and also we want you to be able to use this information to develop effective clinical strategies. But again, the case study will really help us explore that. So starting off with hormone balance. So even though hormones and fertility are very tightly linked and do cross over significantly, the first part of this talk will really focus on the hormones, and then we'll move into the pregnancy section. And the reason I'm doing this is because even though there is a lot of crossover, when someone comes to me and says, my issue is high estrogens, I'm going to pull out different genes. And if someone comes to me and says, I'm interested in preconception care, or I've had a baby with neural tube defects or stillbirth or 6 miscarriages. So yes, as I said in the early modules, we want to have a holistic approach and look at as many things as possible. But if we're going to narrow things down, and we kind of are for the modules, there are certain things I'd look at for hormones and certain things for pregnancy. So hormones are chemical messengers. They're involved in the growth, the metabolism and the reproduction, and are important for reproduction and maintaining homeostasis. Hormones can influence the immune system, the brain, and our behaviour. And the endocrine system is much slower than the nervous system, but it has widespread, long-lasting effects. And it's important to note this because if someone has a genetic variation that perhaps increases enzyme activity and they're more likely to produce more testosterone or more estrogen, that could have a significant impact when it's just tiny amounts of hormones that really do communicate with our body, with our brain, and affect how we feel, our reproductive system, etc. Hormonal imbalances occur over a long period of time and can cause various health outcomes. So PCOS, infertility, mental health disorders and autoimmune conditions such as thyroid, thinking about Hashimoto's or Graves', are all associated with hormonal imbalances. And hormonal imbalances are a reflection of our genes, our lifestyle choices and our environment. So let's look at the genes that influence hormone synthesis, so the production of hormones. So before I sort of show you these particular genes, it's important you understand the cytochrome P450s. We often call them the CYPs or the CYPs. So the cytochrome P450s are genes that code for enzymes that are involved in the synthesis of hormones, which is why we will talk about them, but also cholesterol and other fatty acids. They also metabolize a number of medications and chemicals and toxins, and you will hear about them again in Module 6. There are approximately 60 cytochrome P450 enzymes in humans, and they're actually classified or named a little differently. So when you think about MTHFR, it's methylene tetrahydrofolate reductase. It's the function of the enzyme, it's telling you what you do, and then it's the abbreviation of the name. Or if we had PEMT, phosphatidyl ethanolamine methyltransferase, transcobalamin 2, TCN2. So when we're looking at gene names, there are often these abbreviations that actually are related to the function of that protein or the enzyme or the transporter. With the cytochrome P450s, you'll always see the CYP, and then there will be a number which relates to a specific group that it's associated with. There'll then be a letter representing the subgroup, and then we have another number. So in the example here, you can see CYP27A1. So when you see the cytochrome P450s, they always have this classification or nomenclature. So you have to remember what they do, unlike seeing something like TCN2, if you do remember that that's transcobalamin 2. The cytochrome P450s are highly polymorphic. What I mean here by highly polymorphic is they have many genetic variations that influence the function. We often say that cytochrome P450 enzymes are associated with fast or slow metabolizers, or the fast metabolism of a certain drug or a certain toxin. They contain heme as a cofactor, so iron. They're referred to as hemoproteins. And diseases that are influenced by the cytochrome P450s are generally due to mutations that result in a build-up of reactive compounds, and you'll understand more about that when we go through the detoxification module as well. So focusing on hormones with the cytochrome P450s, if someone comes to you and they're interested in PCOS or higher androgens or estrogens, the cytochrome P450s you want to identify are the CYP17A1 and the CYP19A1. CYP17A1 is the enzyme that pushes pregnenolone and progesterone over towards the androgens. It actually has two functions. So I mentioned in the previous module, and I think I said in the early modules, most genes code for a protein that has a specific function. So in the previous module, I said MTHFD1 was a trifunctional protein or trifunctional enzyme. In this case, CYP17A1 actually has two functions. It has hydroxylase activity. It adds that OH molecule to both progesterone and pregnenolone. So it first converts it to the 17-OH. Then it has lyase activity where it can actually cleave that molecule, resulting in the production of our androgens. So this particular enzyme is essential for the production of our androgens. And there is a genetic variation that's actually associated with increased enzyme activity. So these people are more likely to have higher androgen levels, possibly even higher estrogens, because you're pushing your progesterone and pregnenolone over toward the androgens that can come down to the estrogens. In addition, there's some studies, it's not a lot of evidence, but some studies suggesting that the genetic variation within this gene may also be associated with PCOS, and we know that PCOS is also linked with higher androgens. CYP19A1 that you can see at the bottom is what converts our androgens down to our estrogens. It's also known as aromatase or estrogen synthase, and aromatase is actually heavily influenced by not only the genetic variation that I'll show you, but environmental factors, depending on someone's adipose tissue, alcohol intake and things like that. So CYP17A1, as I said, it's got two functions, hydroxylase and lyase, and it's what pushes your progesterone and pregnenolone over towards the androgens. There's a particular SNP, and it's the G allele, or the C, depending on what strand of the DNA that you are looking at, is associated with increased activity, which can result in higher androgens, higher estrogens, and there's some evidence that it may be linked with PCOS, and there is that reference at the bottom if you want to look at that further. With aromatase, we know this is what converts our androgens to our estrogens. The CYP19A1 is most active in the ovaries, whereas in males it's most active in the adipose tissues. So if your male patients are coming in and they're overweight and they're worried about estrogen, the first thing you can say is, well, as soon as you lose weight, your estrogen levels are going to go down, because that is the tissue. Once upon a time, we thought that fat tissue or adipose tissue was just a storage tissue. Some people call it even an endocrine tissue, because there are metabolic activities going on within that fat tissue. The particular SNP RS146, the T allele, is associated with increased enzyme activity and increased conversion of our androgens to estrogens. So if someone comes in and they're concerned about higher estrogens, think about CYP19A1 or aromatase, and then what you can do, which we'll talk about later in the therapeutics section, to try to compensate for that. So we've talked about some of the genes that actually influence the synthesis of our androgens and estrogens, but we also have genetic variations within genes that influence the metabolism and breakdown of our estrogens that can also influence the levels of our hormones. When you are considering the breakdown or the metabolism of estrogens into the catechol, estrogens or the estrogen metabolites, you're thinking about CYP1A1. CYP1A1 pushes estrogen down the 2-hydroxy pathway. Now, this is thought to be the least harmful. I wouldn't say it's beneficial. I've heard some people say this is sort of the protective estrogen. It's still forming a reactive metabolite, but it's less harmful than the 4-hydroxy. So the 4-hydroxy metabolite, which is shown in red, is actually the most reactive metabolite of estrogen. This has the potential to turn into things known as quinones or semiquinones, and they can cause DNA damage. So it's CYP1B1 that pushes estrogen down to the 4-hydroxy metabolite. So you definitely want to be looking at this with your patients that come in with high estrogens. Are they making the 4-hydroxy metabolite? Because that is the potentially dangerous metabolite. And we've also got the 16-hydroxy metabolite. So it's the 2, 4 and 16, and it's CYP3A4 that converts estrogen to the 16-hydroxy. So CYP3A4, like many of the cytochrome P450s, has multiple functions in that it also metabolises about 50% of the pharmaceutical medications that we can get here in Australia. More research is done on this in relation to medications than with hormones, but it is the enzyme that converts to the 16-hydroxy, and we know that this form can go back up to estrogen. And again, it is reactive and can also link to quinones and potentially DNA damage, but it's the 4 that is shown to be the most reactive. So with cytochrome P450-1A1, which is what pushes estrogen down to that 2-hydroxy pathway, there is a particular SNP at position 2455 where an A changes to a G. The G allele is associated with increased enzyme activity, so this could potentially mean that you would have an increased amount of 2-hydroxy metabolites. That may not be an issue. You need to think about it in relation to your patient. I'd be more concerned about the 4-hydroxy. The G allele may also increase the conversion of that 2-hydroxy metabolite, as I mentioned, but also know that the cytochrome P450-1A1, 1A2, which we'll mention later, particularly in relation to caffeine, and 1B1, yes, they metabolise the estrogens, but they also help us to break down or metabolise what are known as polycyclic aromatic hydrocarbons. So these are your benzenes, your cigarette smoke, burnt meats, things like that, as well as other aromatic amines and reactive intermediates. The G allele has been associated with reduced sperm quality. So yes, it's affecting the estrogen levels, but it may also impact on male fertility. And there seems to be an interaction between this G allele that affects enzyme activity and smoking, which can increase the risk of infertility. When we look at cytochrome P450-1B1 or CYP1B1, there are a number of genetic variations that have been identified in this SNP. As with all of the CYPs, as I said, they're highly polymorphic, but the one that you want to look for when you're looking through the genetic test results is the 432 leucine to valine. So in this case, they report it in the literature as the amino acid. Remember in the early modules, I said sometimes we talk about allele, sometimes we talk about amino acids. In this case, you'll see the L432V in most cases. This is the one that has the most evidence, or I would say the evidence to suggesting that this SNP has the biggest effect on the actual enzyme. So the G allele is associated with increased enzyme activity, and this potentially can increase that conversion and encourage estrogen to go down that 4-hydroxy pathway. And that is that sort of more reactive, potentially dangerous metabolite. The evidence is limited. Now, I say that because I remember first learning about CYP1B1 and thinking there was very, very strong evidence for this. But once you start to look into it, there's not a lot of evidence, and that is because not many papers actually measure the 4-hydroxy metabolite. When you look through the literature, a lot of the metabolites that were measured in the past were the 2 and the 16. They're looking at the ratio there. But most papers don't even look at the metabolites. It's not the interest of the researcher. They're looking at things like breast cancer or health outcomes. So we know that the function is to push it down the 4-hydroxy pathway. We know that enzyme activity is increased, so we are putting that together as the mechanism. So I hope you understand that, because I was a little shocked when I first started reviewing the research that it wasn't as clear as I thought. So definitely the mechanism is correct, but not a lot of evidence around levels because it's just not there. We need someone to sort of, look into that and not just focus on the end outcome of breast cancer, because we want to understand these mechanisms. It also metabolises the environmental pollutants, as does CYP1A1, and it's associated with various cancers, prostate, breast and endometrial. There is a lot of research around CYP1B1 and this particular SNP. It's one of the things that I jump to when someone comes in and says, I've got really high estrogens. What's CYP1B1 doing? Are they making the 4 metabolites? And if so, then what are we going to do about that? And you can obviously measure these as well. So we're looking at the synthesis. We've got our progesterone. It's gone over to our androgens, our estrogens. We're starting to try to metabolise and clear these estrogens. We have those metabolites. Then these metabolites need to be cleared. Remember I said they are reactive as well. So we want to try to reduce the damage that these can do, or reduce the ability to be converted into the quinones and semiquinones. One of the enzymes that does that is COMT, catechol O-methyltransferase. So you've just had a whole lecture on methylation. You're now an expert in methylation, and you'll know that COMT, being a methyltransferase, also then would sort of factor in your MTHFR, your MTHFD1, your BHMT, what's going on with that folate homocysteine pathway. If someone has a SNP and they have a slow COMT, it's usually classified as fast, intermediate or slow, there are a few things you can do. You can actually see it in this diagram. We've got S-adenosyl methionine, so making sure that methylation is adequate, but also magnesium is a cofactor. So we want COMT to add a methyl group onto these catechol estrogens, and we then get these 2-methoxy or 4-methoxy estrogen metabolites, and these are less reactive. This is helping with the breakdown and elimination of these estrogens. You can see that the 4-hydroxy metabolite there, yes, has the potential to go through the COMT. That's what we want. We want to push it through these enzymes to help get it out of the body, but also they can form these quinones, and there is an enzyme, NQO1 or quinone reductase, that can actually reduce these quinones back to the metabolites so that the quinones can't go on and cause DNA damage, because that's what they do. They're very, very reactive. So with COMT, there are 2 versions. There is the membrane-bound COMT, which is mainly in the brain and the nerve cells, and you'll get to hear all about this in the neurotransmitter module. And we also have soluble COMT, which is found in the other tissues, and this is mainly involved in metabolising the estrogens. Again, we're looking at the amino acid. When you're trying to identify the genetic variation that is going to influence the activity of COMT and the ability to metabolise the estrogens, you are looking for the Val158-MET. So this is a valine to a methionine. It's shown as the amino acid. I don't think I've ever looked through the literature and seen it as the nucleotide sequence. No doubt the most evidence is for this. I have people coming to me showing me multiple COMTs, but you just want to focus on this. The other COMT SNPs don't have enough evidence for you really to sit there and try to make a clinical decision or change the way you treat your patient. The GG, which is the Val-Val, is known as the faster COMT. The AG is the Val-MET. This is your heterozygous. And then we have the MET-MET. This is the slower one. So you need to think about that in relation to your patient. I'm not going to talk about the neurotransmitter side of things today. We'll just focus on the estrogens. And in relation to the estrogens, if someone's got high estrogen metabolites and they've got a slow COMT, again, when I said we're going to pull out the CYP1B1, we really want to think about what COMT's doing and try to support that. So you can see here COMT, it requires SAMe. I mentioned that methylation was so important. So this is when you'd start to actually have a look at your patient's methylation profile as well. It also requires magnesium as the cofactor, and there is some evidence suggesting that increased magnesium does actually increase COMT activity in the liver. So the issue with these reactive metabolites, I said that they are potentially dangerous. I say potentially because they may not cause harm, but they do have the ability to be converted into these quinones, and these are very dangerous, very reactive free radicals or compounds that can cause cellular damage or DNA damage and oxidative stress. By causing this DNA damage, that can then go on to be associated with things like breast cancer. So the enzymes that we need to actually help reduce or get rid of these quinones are the NQO1, like I showed you in the diagram, and we'll talk about this in Module 6, as well as the glutathione S transferases. The glutathione transferases are very important, and this is kind of moving into phase 2 detoxification, and you will get the lecture coming up after this on the GSTs and phase 2 detox. So we've looked at particular genes that influence our hormone levels. Someone might be more likely to have higher estrogens or higher androgens depending on the genes they've inherited. But there are also some genes that have been particularly associated with reproductive or hormone disorders such as PCOS. So there have been several genome-wide association studies that have identified a particular SNP with risk for PCOS. And this is the luteinizing hormone choriogonadotropin receptor. Another one you don't want to say out loud, but the LHCGR. So what LHCGR is involved in is testosterone, in producing testosterone in males, and in females it stimulates androgen production. The SNP, which is an A to a G, but depending on where you read, sometimes shows it as a T to a C. Remember I said sometimes we look at the opposite strand. So the A to T increases risk for polycystic ovarian syndrome, so PCOS. There's actually a reference that you can look at there if you're interested in that. So if your patient does come in with this particular SNP and their concern is around, androgens or PCOS, or perhaps they've already got it, you want to start to think about ways to reduce these androgen levels, ways to compensate for this. And again, we'll talk about it in the therapeutic section. In relation to thyroid disorders, there is a particular gene, FOXC1 or FORCAD-BOXC1, also known as thyroid transcription factor, and this is involved in the regulation of thyroid hormones. So there's a particular SNP that is associated with altered thyroid function, and in this case it's actually both the wild type and the variant that can have an effect or has shown to have an effect on health in the published literature. So the G allele has a strong association with hypothyroidism, but also the A allele is associated with thyroid cancer. So for those of you who look at this direct-to-consumer and getting, hundreds of genes, when you look at FOXC1 and you look at this SNP, you'll usually see a red, and they are identifying the cancer. So they focus on the cancer. They don't mention the hypothyroidism. So again, when we're talking about the reds and greens, sometimes it's misleading. The hypothyroidism, there is good evidence for that. It's in association with nutrient deficiencies. So this is where you want to make sure that patients have got enough iodine, selenium, zinc, things like that. And the cancer association is in association with previous infections, radiation exposure, and again, nutrient deficiencies. So what can we do if your patient comes in, you've identified there's some genetic risk factors, they're having concerns or issues with hormones? You need to talk to them about diet. As we always say, diet is key, but in this case, nutrient deficiencies really do influence our hormone levels. We've got to make sure there are no nutrient deficiencies. In relation to things like COMT, if they've got high estrogens and we want to support the metabolism, the excretion of these estrogens, we need to think about methylation, the B vitamins, the folate, what are the genes in that particular pathway. Other things like omega-3s, vitamin A and vitamin D, these are important too for hormones. Again, think back to Module 3 where we went through the nutrients. Some people will require more vitamin D, some people may need more vitamin A. The minerals are important, not just for thyroid, but supporting antioxidant enzymes and overall health. Zinc, when we're talking about, testosterone and estrogen, we know that zinc is very, very important. And there are actually aromatase inhibitors. So if someone's got a genetic variation and they are likely to have a faster aromatase, so they're going to convert more of their androgens to their estrogens, you can compensate with things known as aromatase inhibitors. So cruciferous vegetables, there's lots of research around cruciferous vegetables, slowing down aromatase. So that's the first thing you can tell your patients, include more of these in your diet. Specific mushrooms, catechins and quercetin. And the reference I've got there has actually got lists of aromatase inhibitors, and it's a review and it puts all that information there for you together. When it comes to herbs, Even though I'm not a herbalist or a naturopath, I do use Vitex, so Chase Tree. We know that can help with balancing progesterone and estrogen. Licorice has been shown to block the actions of CYP17A1. So remember when I said CYP17A1 is what pushes your progesterone and pregnenolone over to the androgens? If your patient comes in and they've got high testosterone, high DHEA, high androgens, you can actually slow that enzyme down with licorice, and there's some evidence for that. We need to ensure that our patient's diet, as I said, is good and minimising exposures to chemicals and toxins that can act as endocrine disruptors, because that will also influence the function and the health of our liver. So a lot of these enzymes are actually working in the liver, so we need our liver to actually be functioning properly and to be healthy. For those that do have dysbiosis, they may have high glucuronidase. That can actually result in estrogen being recirculated back into the body. Calcium D-glucarate can help there. And I've mentioned COMT and methylation, but no, that's not the only way to try to help the body support and break down and eliminate estrogens. Glucuronidation is obviously important, but also sulfation. There are drugs, chemicals and toxins. Alcohol increases aromatase. Your patient comes into you and she says, I can't lose weight on my hips, I've got sore boobs, I've got moody, her estrogen's up here, and then she says, I have, 3 glasses of wine each night, it relaxes me. One of the first things you're going to say is that I can actually start to turn down your aromatase. We can do things to bring your estrogen down that's going to help you feel better, hopefully start to help you lose a bit of weight, particularly in these sort of estrogen-dominant areas, but you're going to have to stop your alcohol. She might not like it, but when you can actually start to tell her the mechanism and how it will work, that could be the key to go, you know what? I don't want to drink this alcohol. Same thing in males. They come in, they're telling you about, symptoms, man boobs, things like that. Get rid of the alcohol. Medications. We haven't discussed it yet, but we will in Module 6. These cytochrome P450 enzymes are influenced by chemicals and toxins and medications. Your patient might be on a particular medication that is increasing or decreasing one of these CYPs that is directly influencing their hormone levels, and they may not even know it. They may need to go back and have a chat with their doctor about the medications they're on. Mould is something that can wreak havoc with our hormones, and again, these environmental pollutants. Stress, sleep, circadian rhythm. We all know it, but there's no point coming in and saying, I want you to have your B vitamins for methylation, I want you to take these herbs, I want you to do this, this and this, and then they tell you they're only sleeping 3 hours because they're on their phone and checking emails and doing all these kind of things. It's going to be very, very hard for them to lose weight if they're not sleeping. So we need to get the basics right. They need to sleep, they need to exercise, they need to have a good diet, and then we drill down into the genes and give these sort of personalised recommendations to help them achieve these results. So moving on to fertility and pregnancy health. So fertility complications are increasing in Australia and around the world. The use of assisted reproductive technology, or ART, and IVF in developing countries is rising. I think we all know that. We all know someone who has had IVF or is struggling to fall pregnant. Female fertility peaks in our 20s, declines in our 30s and our 40s, and sperm concentrations and semen quality have been decreasing over the past several decades. Doesn't sound too good, does it? People are leaving pregnancy till later, and men are basically providing not very good samples anymore. So 40 to 50% of infertility cases are due to male factor. And this is usually due to low sperm concentration, poor motility and abnormal morphology. So they're not swimming properly, there's not enough of the sperm cells, or there are issues, it has multiple heads, or there's problems with that morphology. One of the biggest causes for this is oxidative stress, exposures to chemicals, toxins, pesticides, things like that, resulting in high oxidative stress. And as I say on the slide, sperm cells are particularly susceptible to reactive oxygen species. This is the oxidative stress, and we will talk about this. Fertility in both males and females are a reflection of our genes, our lifestyle choices, our environment. You heard me say that at the beginning of the hormone section, so it's the same, but also our health and hormonal status influences our reproductive health and capacity. Genetics are related to our hormones, our nutrients, and our methylation cycle, as well as our detoxification and antioxidant capacity. So we have genetic variations that influence all of these. So you've already had a lecture on methylation. We spoke a lot about folate, but what I didn't do was spend much time on epigenetics. And the reason why I didn't spend much time on epigenetics is because I wanted to address that here. So methylation is one of the main epigenetic marks that's involved in turning genes off and on. So in this diagram, we can see there are these CPG islands. A CPG island is where we have a lot of C's and G's in that sort of DNA strand. So you've got your gene, you expect to see a certain frequency of nucleotides. If you see lots of C's and G's in a close region, that's called a CPG island. Those CPG islands are actually what are usually methylated because it's the cytosines, the C's that are methylated. So what you can see here in the bottom is that this CPG island is methylated, shown by those blue circles. Having these methyl groups sitting on the DNA actually blocks the transcription factors that are needed to bind and turn genes on. So basically they're blocking the expression. The gene can't be turned on, therefore it's not expressed. You're not going to make the protein. You're not going to make the hormone, the enzyme, or whatever it's coding for. So it blocks gene expression. It regulates what can be turned on and what can be turned off. And that's very, very important when you consider that every single cell in the body contains the entire genome. So if you're doing genetic testing, you can give blood, you can give a cheek swab, you can give saliva, you could give a skin cell. But these cells need to know how to function. They need to know what genes to turn on. Am I a skin cell? Am I a liver cell? Am I doing something else in the body? Methylation, epigenetics, is what regulates what genes are turned off and on. So it's very, very important. So methylation patterns, these epigenetic marks that are regulating what genes are turned off and on, are inherited. So basically in utero they're laid down, as I will explain, and then once we're born and we grow into adulthood and we age, these methylation patterns should be maintained. And I'm trying to represent that in this image here. So what you can see is two DNA strands. So just this is your DNA helix, not shown as a helix, just two strands of DNA. And you can see the methylation patterns. You've got these methyl groups stuck on. What happens is there's been DNA replication, the cell's replicating, so it's going off to make two daughter cells, and we can see that one of the strands, the initial strand, is still methylated, but that new DNA strand, so it's been replicated, is not methylated, and then has to be methylated. And what should happen is these methyl groups should be stuck on in the exact same place. So if you're making new cells, these methylation patterns should be replicated in the new cells. I was taught that these methylation patterns initially are very, very robust, and generally they are passed on as we age. What we now know is there is something called epigenetic drift, and that these methylation patterns actually do change as we age. We know that when these methylation patterns change, it increases risk for cancer, for neurological disease, particularly things like Alzheimer's. And the things that influence these methylation patterns changing are environmental toxins. BPA is a hypomethylator. I'll discuss that. There are a lot of environmental chemicals and toxins, stress, nutrient deficiencies. I'm hoping that you're already in your mind, you're going, oh, folate, B12, B6, MTHFR, all of these things. They influence the ability to make SAMe, which provides all the methyl groups in the body. So if someone's having a problem with methylation, they're then going to have an issue with methylating all of the DNA during that cell replication process. So what we once thought is that these methylation patterns were inherited. We now know that they can change, particularly as we age, under certain stresses. So I've told you about methylation and epigenetic drift, but the focus of this talk is really around fertility and pregnancy. And the reason I wanted to address epigenetics here is because the in utero environment, so during pregnancy, is so important. When an egg is fertilised, we have the zygote. So in the zygote, we have global demethylation. What that means is all the methylation marks are gone. So the sperm sample or the sperm cell and the egg cell have particular methylation patterns. They come together. These patterns are totally wiped. It's called global demethylation. What happens is the zygote then implants into the uterus, and as the cell replicates and it grows, we actually get global remethylation. So the methylation marks are wiped and then they're put back on. So there is a critical window where that mother needs to ensure that her nutrition is adequate to supply those methyl groups. She needs to be sure that she's not going to be exposed to chemicals and toxins that are going to influence her methylation patterns. And also we know that stress, trauma can influence methylation as well. So because if there is a change at that time when that methylation pattern is laid down, that will then continue into adulthood. And studies have shown, though it's mouse model, it's hard to do in humans, but studies have shown that when we actually have these abnormal methylation patterns or these changes, they can last up to 3 generations. Many of you have probably heard about this. The phenomena is fetal programming. The David Barker hypothesis, he was one of the first to discuss this, showing how mothers that are malnourished go on to have more obese children. As adults, they're at risk for cardiovascular disease, diabetes. These are epigenetic changes due to deficiencies. And we now know a lot more about the chemicals, the toxins, the genes that play a role here. So the environment in utero can alter the development of the fetus, which has a permanent effect. So it affects, the individual in their adult life. Fetal program can be altered by subtle changes due to maternal nutrition, stress and exposures. And this recent review here actually, which was published last year, 2018, actually goes through all of this. It's a fantastic paper if you're interested in this. As I mentioned, David Barker was really the first person to start to make us aware of this. And there is the DOHAD group. It's a group that I used to be with, the Development Origins of Health and Disease. They do a lot of research around methylation, epigenetics, and understanding how our environment really influences that embryo and growing fetus and the effects it has in adult life. So what impacts on methylation or epigenetics? It's our genes and our environment. So it's the diet and the nutrient intake. Of course, we're talking about, B vitamins and folate, but it's not just the Bs and the folate. There are other nutrients that directly impact on sort of epigenetics and methylation, even things like sulforaphane. We'll come into that in the detoxification module. Inherited genetic variations and SNPs. We know that things like MTHFR can impact on methylation because it increases risk for higher homocysteine. We're getting less methionine, less S-adenosyl methionine, less methylation. Exposures to chemicals and toxins, perceived stress and trauma, exercise. Exercise changes the methylation patterns in muscles and things like that. So your environment directly influences your epigenetics. Sleep also influences the methylation patterns. And epigenetic modifications, as I mentioned, have been shown to last over several generations if the changes happen in utero. And it's just in that specific time point. So there's a global demethylation and then remethylation. So if there are some serious stresses at that time point, that will last through to adulthood. So we're not going to go through this again, but just remember these are the genes. This is the metabolic cycle you want to look at if you want to focus on balancing methylation, trying to optimise your patient's health so that you can actually help them try to ensure that these methylation patterns, particularly in early pregnancy, are laid down as they should be. So we know that folate, B vitamins and things like that have a really big impact on methylation. But just to give you an example of the toxins and the effects that these can have on epigenetic regulation, I've used this particular image here. So this is a picture from Waterland and Jertel. Back in 2003, they published a landmark article showing that maternal diet, particularly folate and B vitamins or high levels of methyl donors, can influence coat colour and disease. And in this case, these are sort of follow-on studies. They've shown that chemicals such as BPA can change the methylation patterns in utero. So in this photo, we've got 2 genetically identical mice, and they are the same age. Both of the mothers during pregnancy were exposed to BPA, which is a hypomethylator. Now, most of us know that BPA has been taken out of the plastic, but there are many other chemicals in the plastic that still potentially are dangerous. It's just that we have so much research on BPA now, hard evidence that they've had to remove that in relation to developmental defects, epigenetic changes and cancer. So these mothers have been exposed to BPA. The mother, when we're looking at this yellow, overweight, obese mouse, this mother was on a normal chow diet. The brown mouse, actually the mother had a diet that was higher in methyl donors, more folate, more B12, more choline. So basically by providing all of these methyl donors, It's actually helped compensate for the BPA. So the BPA is a hypomethylator, reduces methylation, can change the epigenetic patterns, but then by compensating with the diet, it's actually helped protect from these epigenetic changes. I am definitely not suggesting that all women in pregnancy have high-dose B vitamins or methyl donors, but in this case, it has helped compensate for these toxins and chemicals. So really the whole purpose of that was just to show you the effects you can have, because this was actually just one gene. If I just go back, this is only one gene that is methylated differently. And by this change in one gene, the methylation pattern has changed in one gene, we now have a different coat colour for life. The mouse is now obese, despite being on a similar diet, and it's likely to develop obesity, diabetes and cardiovascular disease. All these changes just from that slight alteration in the methylation pattern. So moving on to fertility, I've talked about epigenetics, the effect that this can have in utero and adult onset diseases. We know that methylation abnormalities may also be an underlying cause for some fertility cases and pregnancy complications. When we look at the research, often what people are testing is actually methylation, these methyl groups sitting on the DNA, or they're looking at changes in gene expression. In clinic, when you're working with your patients and your clients, we can't actually do that. These tests are not clinically available. So what you want to be looking at is obviously the genes. Are they at risk for having higher homocysteine or methylation problems? But also things like, the folate levels, the B12 levels, what's their homocysteine. You can even look at the S-adenosyl methionine or S-adenosyl homocysteine, that SAMSAR ratio. So when it comes to the evidence and the literature, there is a lot with folate and pregnancy complications, with B vitamins, homocysteine in the genes. There's not so much with the SAMSAR ratio, but I think that's something that's relatively new that's being brought into sort of clinical testing. But it is a good way to try to estimate or determine what someone's methylation status is. So MTHFR is likely to be the most studied gene in relation to fertility and pregnancy outcomes. We discussed it in the last module. We know these genetic variations have a big impact on MTHFR activity, which can impact on an individual's needs for folate, which then directly impacts on methylation. But it's not just MTHFR. You want to look at the MTRR, the MTR. Think about their B12 levels. MTHFD1. This has also been associated with pregnancy complications. And these genetic variations within the methylation cycle are common. This is why we test them, because many people do have them. And they indicate that a significant proportion of individuals have a higher requirement for folate and B vitamins. And that's when you want to come in and start to target and personalise the supplements that your patient may need if they've come in and had serious issues, as in, 4+ miscarriages, or they can't get pregnant. Let's try to pinpoint exactly what they need and optimise health. So in relation to MTHFR, we know that definitely has the most evidence. In 2015, the World Health Organization published a document in relation to the main determinants of folate status, and what they state is that intake, antifolate medication and genetic background, including MTHFR 677, are the main determinants for folate status. It also suggests that the need for folate and B vitamins may be increased depending on the genotype, obviously the MTHFR 677, geographical and environmental differences. So if your patient comes to you and you think they might need higher folate, you can be pretty confident in suggesting that they need higher folate. And I'm not saying just jump to supplements, talk to them about the importance of diet, because even the World Health Organization has acknowledged this, and the NHMRC. So for the dietitians, looking at the dietitians in the audience, and for the dietitians or nutritionists, and even naturopaths and integrative doctors, we're working with nutrition. The NHMRC, the nutrient reference values for Australia and New Zealand state that folate requirements can be affected by bioavailability, so the absorption, nutrient interactions, smoking, certain drugs and genetic variations, notably the 677T polymorphism that causes the MTHF, so methylene tetrahydrofolate reductase deficiency. I feel that the percentage, the frequency of this SNP is underrepresented here. Perhaps it's because I'm working with people that do have complications, but I definitely see it, I think it's a bit more common than what's stated here. But basically it says that it is likely that individuals who are homozygous for this polymorphism may have a higher requirement for folate. So you've got the NHMRC and the WHO. So if you decide you want to give higher folate, or you want to consider increasing doses of supplements, you've got these kind of references to go back to. And really what it's doing is it's emphasising the importance of these genes, because sometimes we say, oh, MTHFR, or I'm not sure about MTHFD1, but when you've got government organisations and the WHO acknowledging these, you really do know that they have, pulled together that evidence and they do have a strong impact. So anencephaly and spina bifida together are referred to as neural tube defects. They occur in 1 in every 500 to 1,000 pregnancies. They are serious and relatively common. So we know that the folic acid fortification that I spoke about in the last module is really to prevent neural tube defects. And that all came about because of this study. So in the UK, the Medical Research Council developed a double-blind, randomised controlled trial evaluating the effectiveness of folic acid with or without other vitamins and the rate of neural tube defects in women who had already had a baby with neural tube defects. And this is because if you've had a baby with neural tube defects, the frequency of having another baby is much higher. What they found is the prevalence of neural tube defects with folic acid was 1% compared to 3.5% in those that did not have folic acid. So there was a reduction in the recurrence of neural tube defects by 72%, and the effect of other vitamins, according to this study, on the risk of neural tube defects was non-significant. At the same time that this study was going on, there was also a randomised controlled trial in Hungary, and it looked at a multivitamin in relation to neural tube defects, but first occurring, which is obviously more rare. There were no neural tube defects among the 2,000 pregnancies in the multivitamin and multimineral study, and the protective effect of the multivitamin, which did include 800 micrograms or 0.8 milligrams of folic acid on the first occurrence of neural tube defects, was estimated to be 90%. So it's huge. So if people are having folic acid, or in this case, I even think a multivitamin, that reduction in neural tube defects is quite effective. And for those of you who do look into this, you would know that the Australian Institute of Health and Welfare in 2016 produced a document, because they are monitoring the rates and frequencies of neural tube defects since the introduction of folic acid fortification in Australia. I think as a whole, it was definitely less than 20%. I think it's about 16%. As a whole, there's been about a 16% reduction in neural tube defects in Australia, but in teenagers, so in teenagers that fall pregnant, there is a reduction of 55% in the cases of neural tube defects. And when it comes to Aboriginals and Torres Strait Islanders, I think it's a reduction of over 70%. So I think we need to be focusing on real foods, gut health and diet. But when we, have these kind of statistics, we can see why there is this fortification. So over 80 countries have introduced mandatory fortification. It's believed that the number of neural tube defects has decreased by up to half, and that adequate maternal folate has shown to reduce the risk of other birth defects. So we focus on neural tube defects because that's where the hard evidence lies. However, we know that folate also reduces the risk of cleft palate and other congenital abnormalities. And as I said in the last module, that the folate expert groups indicates there's no evidence of harm, but I think we all need to keep an open mind. We all need to keep following the evidence, educating our patients about real food as well, because it's the people that are lacking the nutrients that are deficient that are actually getting the benefits. So that's the issue. These people aren't getting enough folate from their diet. Smoking affects folate. People that smoke have lower levels of folate. It breaks it down. So, talking about smoking in communities where they're likely to be smoking, more education around these things as well. So when we talk about the optimal levels, if you go to your doctor and say, I want to fall pregnant, what level of folate do I need to reduce my risk of neural tube defects? We don't have a level pre-pregnancy. We don't have a level pre-pregnancy because most of the research around folate, neural tube defects and pregnancy disorders occurs when women are pregnant. So they'll come in and have their bloods taken when they're pregnant. So we can't sort of say, well, if you want to fall pregnant, you need to have a red blood cell folate of over 1,200 nanomole, but I'm just throwing that out there. But what we do know is that at the booking bloods, so in Australia All women come in for that first trimester booking bloods, we call it, and it's usually around sort of 10 to 12 weeks. And we know that if women have a red blood cell folate level above 900 nanomole, 906 to be exact, nanomole per litre, that is associated with the lowest risk for neural tube defects. The WHO also states this. It's based on a paper that's in this slide. We know that plasma and red blood cell folate decrease during pregnancy. So because we know it decreases during pregnancy, we have very, very good evidence for that. And not just in pregnancy, it continues to decrease during breastfeeding. For women who are not having an adequate nutrient intake or are not supplementing in late gestation or in breastfeeding, folate levels continue to go down. And even if a woman is, or the mother is getting towards folate deficiency, she will still preferentially direct that folate through the breast milk to the growing baby. Great for the baby, not so good for the mother. So plasma and red blood cell folate decrease during pregnancy, which is why I suggested a higher level pre-pregnancy, because we know it's going to come down. Some studies, so getting back to the question I was asked at the end of Module 4, some studies have suggested that 5-methyl tetrahydrate may even be better at increasing levels of folic acid. I don't think we have enough evidence yet to make that statement, but we have some emerging evidence suggesting that it may be a better form. In relation to folic acid during pregnancy, there has been some studies suggesting there could be concerns in relation to asthma. So there was a study in Adelaide that found that women who were supplementing in late gestation, so 30 to 34 weeks, were more likely to have a child who developed asthma at the age of 3.5 years of age. However, they no longer had asthma at 5.5 years of age. So how you explain that, I'm unsure. The authors state that it may be due to epigenetic regulation. There are some comments on this paper for anyone who wants to look into that. The reason why I've got this paper in here is because sometimes patients will come in and ask, and also if you remember when this paper was published, I do, I was working in Adelaide, I know the authors. It was in the media, everyone talked about it for quite some time, and a particular company even brought out a supplement. I don't know if anyone remembers this. There was a company that brought out a first, second and third trimester pregnancy supplement, and they actually didn't have folate in the third trimester supplement, and they quote this paper. So whether this paper is true or not true, it's an epidemiological study. They've gone back, looked at the notes. It's still only one paper. And when you think about the importance of folate, and what I tell people is the recommended dietary intake for all of these nutrients is throughout pregnancy. It doesn't all of a sudden in the third trimester drop. So you need to ensure that your pregnant women, not just as the baby is developing, not just so that they have a healthy child, but when think about adult onset diseases, they are getting adequate nutrition throughout pregnancy and even breastfeeding as well. That's my opinion, but you definitely, if you get asked about this particular paper, you can say that you are aware of it. But as I said, the recommended dietary intakes do not change, and if someone's not meeting it, they need to think about supplementing throughout pregnancy. So autism is a really big concern, and studies have indicated that higher dose folic acid may increase the risk for developing a child with autism. There's also been suggestions that the folic acid fortification program may be associated with this increased frequency of autism. So I haven't seen the data. We do know that these things are being monitored. It would be interesting to try to find out that information, because we do know that autism is increasing. In contrast to this, though, I need to mention there was a study that showed, or multiple studies showed that folic acid may reduce autism. However, these are in mothers that are exposed to high levels of chemicals and toxins and on anti-epileptic medication, which we know reduces folate. So even think back to that mouse model study I showed you. The higher levels of folate and methyl donors actually help protect against these things that were hypomethylators. If someone's having something that's reducing methylation, maybe in this case it's a good thing. There are studies showing that folinic acid improves verbal communication in children with autism and language impairment. And there was a randomised control trial, double-blind, placebo-controlled, very good study suggesting that folinic acid may be a benefit. So if your patient comes to you and they're asking about this, perhaps you can talk about folinic acid. Again, I am saying this to you because the type of people that are actually requesting genetic testing that come to you and say, I want MTHFR tested, and you say, no, I think you need to do a full panel because we want to look more than MTHFR, or they're coming to you with direct-to-consumer testing with hundreds of genes. Many of these people, well, in my world anyway, a lot of these are parents and families of children with autism that want to do whatever they can to try to reduce the risk of autism in, with future children. So genetic testing is very common in the US. We know that autism is also common in the US, and there are a lot of sort of information sites about genetics and autism and MTHFR and methylation. So think back to these papers. If they're asking about these things, you can say, yes, there's some research here or there. Perhaps consider folinic acid. There's some evidence for that. So I'm talking a lot about supplements again, and unfortunately the research on dietary folate is quite scarce. So we want to tell people to eat, real food. There is some studies, and I've mentioned them here. The literature shows that increased food folate is associated with better folate markers. So we're talking about, your levels of folate, and that these levels of folate may prevent neural tube defects because it's improving folate status. But there's mixed reports. There was a study that suggested increasing folate-rich food was actually better at increasing folate and reducing homocysteine than supplements. But even though the food folate was better at reducing homocysteine, it got folate up a little quicker, the food folates did not reach that cutoff of over 900 nanomole in the red blood cell folate. But again, you've had a whole lecture on methylation, and I want you to think about the way that we estimate these things. When we talk about measuring folate, we're also looking at total folate. What would be interesting is having these studies and looking at the amount of 5-methyl, 5,10-methylene, tetrahydrofolate, and how much folic acid is in there. Because remember, the folic acid isn't actually active. So when you're measuring total folate and someone's having folic acid, it's likely that you're actually measuring a lot of that folic acid, terol-glutamic acid, and not the active forms. That's why I was trying to give you so much information on the folate so you can think like that. I think these studies are coming. They're already starting. People are asking these questions. So folate is heat and light sensitive. The effect of cooking is really important. Think back to when I know some parents, when we were young, over boiling our vegetables, really you're degrading and breaking down these important nutrients, including folate. Steaming is best for maintaining that nutrient value, particularly with folate that does break down. And if your patient can tolerate even thinking about eating some raw veggies, depends on how good their gut health is. Folate does not work on its own. There is so much research on just folate. I don't want you to think I'm saying that folate is the most important, but that's just actually where the research is, so I'm reporting to you what's out there. We still need to think about the B12, the B6, the B2, these cofactors, so that folate can do what it's meant to do, go around that methylation cycle, make the DNA, provide the methyl groups. There are no clinical trials on the effectiveness of 5-methyl tetrahydrofolate in preventing neural tube defects. And what I mean here is that I showed you the two studies, or in particular that UK study, that looked at folic acid against nothing in preventing neural tube defects. We couldn't now run a trial looking at 5-methyl and nothing and looking and to see if 5-methyl perhaps reduced risk by 80%, unlike the folic acid that brought it down by 72%. That would be unethical because we know that folic acid is important for pregnancy. So we're not going to get those studies. The only way that we really could start to confidently say that 5-methyl tetrahydrofolate is better, or perhaps something like folinic acid is better than folic acid, is by having some really large studies, thousands of pregnant women, following them over time and then comparing. But this would cost a lot of money, and whether the people that fund this kind of research think this is justified, at this point we're unsure. But that's what we need. So the question that I was asked at the end of Module 4, and that I'm asked all the time, is 5-methyl better, or can we use this, why can't we use that? At the end of the day, the folic acid research has very, very strong associations in reducing risk of neural tube defects, and that's what people focus on. But I do believe there's enough now showing that 5-methyl is safe, folinic acid is safe, And I'm not anti-folic acid. I think a little bit is fine. It's just when you're getting to those... Some people need higher doses. You've seen that from the genetics. The WHO say it, NHMRC says it. You understand the genetics now. Some people need more. For your patients that need more, it's worth considering the natural forms because then they don't get the unmetabolised. And if there are some potential health effects, and there may be, you're avoiding that. So metabolic studies have shown that 5-methyl tetrahydrofolate is biologically active. It seems to be at least as effective as folic acid improving folate biomarkers, and supplementing with 5-methyl tetrahydrofolate for neural tube defects prevention seems to be rational. And there is a nice paper there that goes through all of this information. And as I mentioned, if your patients are coming to you and they've got all of these genetic variations in methylation, you need to support methylation and their concern is autism, think about things like folinic acid, hydroxycobalamin and things like that. So SNPs in the methylation pathway predispose to a high risk of birth defects, pregnancy complications, particularly when the mother's folate and B vitamins are low. Certain genetic variations can limit the availability of these active forms of folate, so directly supplementing with these natural forms has advantages. The MTHF, or the 5-methyl tetrahydrofolate, does not need to be reduced by dihydrofolate reductase, and folate needs to be given with other B vitamin cofactors so that it works appropriately. So the other thing that is very, very important when we're thinking about pregnancy health and fertility is the chemicals and toxins in our environment. So there is convincing evidence indicating that various environmental pollutants and toxins influence fertility, fetal development and lifelong health. Genetically, many of us are more susceptible because of inherited genes that increase the risk of a build-up of these potentially harmful chemicals because we cannot excrete them properly. We can't metabolise them and then eliminate them from the body. The focus for this really is Module 6. So you're going to get detoxification. We'll talk about Phase 1 detox, Phase 2, and then in Module 7 we've got oxidative stress. But I still do need to just highlight it here because it is so important. If your patient comes to you and they're interested in preconception care, and if they're a male with abnormal semen parameters, we want to be talking about this. So oxidative stress is influenced by free radicals, things that are going to increase oxidative stress, so pro-oxidants, and then we have our antioxidants. When it comes to the genes, I've already briefly mentioned them in Module 3, superoxide dismutase, catalase, GPX1. People have genetic variations in these that can reduce enzyme activity, and these people do not break down these free radicals as effectively. So they're at higher risk for oxidative stress. And of course, with that, we need to ensure that our antioxidants from the diet are good as well. So a sperm cell is very susceptible to oxidative stress. It can come from environmental pollutants, drugs, smoking or pollution, but also metabolic disorders. Endogenous free radicals, endogenous oxidative stress can also affect the sperm cell, affecting the numbers, affecting the way that it can swim and the morphology. You've seen this slide before. We're talking about the impact that You've seen this slide before. We're looking at the increasing protective effect of antioxidants, and what you can see at the top of this pyramid are those antioxidant enzymes. These are your primary endogenous antioxidants. They are very, very important for balancing oxidative stress. So MN-SOD, or superoxide dismutase, is the enzyme that converts superoxide. This is very reactive, very dangerous, can increase risk or increase oxidative stress significantly, damaging cell membranes, damaging DNA. There is a particular SNP, RS4880, that has been shown to reduce enzyme activity, so someone's ability to break down superoxide is reduced. And then we also have GPX1, glutathione peroxidase and catalase. And both of these enzymes are important for breaking down hydrogen peroxide, totally neutralising that free radical to oxygen and water. So there are SNPs in both of these, again, that can reduce enzyme activity. And as you can see here, GPX1, it's important in the placenta, and also catalase being in the sperm cells. So directly trying to combat oxidative stress in these sperm cells. This diagram is just showing you the breakdown of these free radicals. So the superoxide is converted by SOD to hydrogen peroxide, which is still actually a free radical and itself can cause oxidative stress, but it's not as potentially damaging as a superoxide, not as reactive as a superoxide. And then it's catalase and GPX1 that break these down to water and oxygen, so totally neutralising these free radicals. If your patient comes in, as I said, they're looking at preconception care, in particular if it's the male and we're talking about abnormal semen parameters, there is a lot we can do to try to reduce oxidative stress. When we talk about these particular genetic variations, now I'm touching on them lightly because you heard them in Module 3 and you're going to get a whole Module 7 on oxidative stress and antioxidants. But it's manganese that is the cofactor for manganese superoxide dismutase. You need to ensure there's adequate manganese. Glutathione peroxidase requires selenium. It's a selenoprotein. If someone's low in selenium, that glutathione peroxidase, which also requires glutathione, is not going to function as well as it should. And with catalase, it requires iron. So we need to think about the minerals. You need to think about the other antioxidants. When you hear people say, eat the rainbow, why do we say eat the rainbow? Because of all the different antioxidants that come in the purples and the greens and the oranges and the yellows and the reds. I'm talking about diet and I'm talking about supplements, but as we can see, lifestyle. Number 1, reduce exposure. A male comes in and you really need to say to them, you need to stop smoking. Or they are exposed to things like, benzenes or, environmental pollutants, educating them on the effects that it can have in relation to oxidative stress and potentially semen health. Also, nuts and fatty acids are important. And when it comes to supplements, there are the vitamin C, the glutathione, the NAC, there are so many supplements. You need to think about what is the right supplement for your patient if you need to support this. I'm not saying everyone needs supplements, but there really is a lot when it comes to supporting oxidative stress, a lot of antioxidants that can help combat these free radicals. So in summary, hormones, fertility and pregnancy outcomes are influenced by our genes and the environment. Methylation and oxidative stress are often underlying causes that you can address. When we talk about hormonal health, if someone comes into you and their concern is the androgens, testosterone, DHEA, or things like PCOS, look for that CYP17A1, look for the LHCGR, and then determine, well, is it a genetic thing? If it's not a genetic thing, what are their exposures? Phthalates, parabens, all of these things that we'll discuss in the next module. As I said, weight is important too. So identifying these genetic variations, if it is a genetic thing, we know that with CYP17A1, you can start to downregulate that. Think about the licorice. With the LHCGR, other things we can do to help regulate our testosterone and estrogen, things like zinc. With our estrogens, there are a lot of genes you can look at to try to identify someone's genetic predispositions for higher estrogen. Do they have the aromatase? Are they converting more of the testosterone to estrogen? Have they got CYP1A1? Are they likely to go down the 2-hydroxy or the 4-hydroxy? What's the COMT, which is involved in methylating these reactive estrogen metabolites? NQO1 for the quinones and GSTP1. So if someone comes to me and their concern is high estrogens, because of all of the symptoms associated with estrogen dominance, or they've actually got levels to tell me that they've got high estrogens, I'm going to be thinking, I already know they've got high estrogen, whether it's due to aromatase or not, but I'll be thinking, is it CYP1B1? Have you got a faster CYP1B1? Are you going to make more 4-hydroxy? Have you got a slow COMT? Are you going to struggle to break down those metabolites? Have you got NQO1? I haven't mentioned NQO1 in detail here because I don't want to overlap too much. You're going to get more of that in detox, but just know that if you inherit the homozygous variant for NQO1, it's associated with no function at all. That a person's ability to reduce those quinones, it's not happening. So they're going to have to think about things like, the glutathione pathways here. So I'm going to identify those. Of course, you can measure the metabolites, but then I'm going to try to address those. With NQO1, we know that there's CoQ10. Again, we'll talk about it in the next module. But also with estrogen, there's a lot you can do lifestyle-wise. As I said, the alcohol, reduced exposures. There are a lot of endocrine disruptors that mimic estrogen. These need to be removed from the environment. Get rid of the plastics. Get rid of some of these skin creams, with parabens and preservatives that act as endocrine disruptors. With fertility and pregnancy health, methylation, you've heard a lot about it, antioxidants, thinking about these genes that are involved in breaking down these free radicals. Does your patient have genetic variations in these antioxidant enzymes, making them more susceptible to oxidative stress? If they do, they need to be careful. They need to minimise exposure. They need to support these enzymes. Consider more antioxidants in the diet. Individuals who inherit certain genes may require a higher intake of folate and B vitamins. We went through that in detail. And finally, what I would like to say is that preconception care, so couples coming in to optimise their health to have a child, may increase natural as well as assisted, so IVF, pregnancy success and optimise fetal outcomes. But what I also want to mention is it's not just the fetal outcomes, it's lifelong health, reducing risk of diabetes, obesity, Alzheimer's, dementia. And that's what we want to talk to our patients as well too. It's not just get them pregnant, let's talk about lifelong health for the parents and that potential child. And of course, balancing hormones comes into that too. So that's Module 5, and I'll see you again for Module 6.